Well, thank you for being here, and I have to say it's exciting for me to both be with amazing scientists who've been studying oaks for many, many years, and also amazing individuals who've been working, studying the natural history, collecting, doing all sorts of things to appreciate this amazing taxa. So I'm an oak heaven, and the work I'm going to do is basically what I need to do. I focus on the population level, and many, many species of oaks I've worked on in California and in elsewhere. I've worked on Quercus humboldtii. I've worked on species in eastern U.S. But I always take a population approach. But as I've looked more and more into the evolutionary biology of oaks in California, in valley oak in particular, it was clear to me I needed to understand the phylogeny, the genetic basis of traits, and become more and more involved in using genomics as a tool to understand how genes allow oaks to grow where they grow. And for this study, I have many collaborators, particularly Joaquin Ortego, who several years ago, came in as a postdoc in my lab, and we continue to collaborate, and he shares an interest in landscape genetics and phylogeny of oaks in California. Okay, so then there's other people, key people, and then I'm going to go on. There's really a cast of thousands because I just, over the years, keep working with whoever comes to my lab anywhere from a month to 6 months and has nothing to do. I help them out. But for this project, Aaron Riordan, who's done some of the niche modeling, Jinjiang Wei, who's doing the phylogenetic work with me now, Paul Guger, who's been my postdoc for years, focusing on the other parts of the work, but certainly involved in this research. And more recently, Sorrel Fitzgibbon, the bioinformatician, and if you're doing genomics and you don't have a good bioinformatician, you are in deep trouble. So she's really helped just escalate the work in the last year that's going on in our lab. So why am I talking about this particular element? And I really have several questions. One is I want to understand the role of integration and species relationships in oaks in general, and particularly how it introduces genes for local populations to grow. California is a particularly wonderful environment because it has environmental heterogeneity, so that over short distances you have species next to each other occupying different habitats and therefore having genes evolve that can spread. And I think white oaks in particular are very, very useful to try to focus on these questions. They have lots of overlapping and separate distributions across this environmental heterogeneous environment. So we could start asking the question, what's the role of reproductive isolation caused by perhaps geographic barriers and ecological isolation in speciation in oaks? And then I'm going to focus a lot today on shrub oaks in California. Everyone's been saying they're a mess, and I thought, if we could just get to the genomic level, it'll clear up. And I'm hoping to give you an idea of what's the answer to that question. So this is where you start seeing the minor glitches that you get when you go from a Macintosh to, I do know how to count. Those go 1, 2, 3, 4. But now I know you're all awake, because you know what, you're an alert audience. Any mistake we have in our slides, you always notice. So lest we think you're not paying attention. Anyway, I'm going to first just quickly introduce the white oaks of California. One would never do such a species-heavy talk except to a group of people that have incredible knowledge of it, but perhaps you'll get overloaded by the end of the symposium. Then we'll talk about the phylogeny of shrub oaks, where we're using microsatellite markers, and then I'm going to go talk about the phylogeny of all the California white oaks, or at least most of them, using DNA sequences. These two markers offer a big contrast in how you can look at relationships among species, and I think as you see the output, you'll get an idea of the contrast you get with those information. Then I want to do is go into a little more deeply with one case study we've been working on for years on hybridization and integration in 3 very Southern California oak species. We'll do a little morphology, and nuclear genes, and then we'll do RNA sequence data. And I appreciate that Paul gave his talk and gave you lots of pictures, because it's probably what most of you would prefer to see. But I look at oaks, although I love their morphology, I love their aesthetics, but I actually look at them from the inside out. So you're going to get very few pictures in this talk. Okay, so let me just introduce the trees and shrub oaks of California with all due apologies to those of you who know them well. So, 4 major tree oak species. I, of course, think that Quercus lobata valley oak is probably the iconic signature species of California that deserves all due respect. And it's got a distribution around the California Great Basin. It typically is in oak savanna habitats or riparian forests. And it has lobed leaves. Quercus garryana occurs north of it, and it goes into Oregon and Washington, and it's an oak savanna and riparian forest tree, and it also has lobed leaves. Then we get to Quercus douglasii, or the blue oak, which is in a woodland, and it's high dense forests, and the leaves tend to be more entire and shallow lobed, so that's a big difference. And then Engelmann oak is in Southern California, and this is not native to California. This originally came up from Mexico and is more related to the Mexican oak species and is very, very different. However, in terms of its ecology, it's a lot more similar to blue oak. No relationship whatsoever. And when you look at the distribution of these species, you can see if you just pull all the arborescent trees, there's a lot of biomass, and Jean made that point very nicely, but look at how much biomass there is in California. You get Quercus garryana right up here and then continuing, and then down here in this area is Engelmann oak. And douglasii, you can see, has its high density surrounding the ring of the Central Valley. And valley oak is very, very sparse. It's not as abundant in high density anyway because of it's oak savanna. However, it's lost enormous amount of its biomass since the Europeans arrived, because it lives where humans want to develop, where we want to build pastures, ranches, suburban houses, wineries. And humans really compete for the same land. So it's suffered enormously. And then agrifolia, which is a red, is another major, and it's only along the coast, and it's not one that I'll be talking about. Now the shrub oaks. The first one I mentioned is Arboretum, but they're alphabetically arranged. It's the California scrub oak. It's the most widespread oak all over. I'll show you some distributions next. Cornelius mulleri, the Muller oak. Down in the deserts. Quercus domosus, the coastal scrub oak. Quercus dorata, variety dorata, it's called leather oak, and it's restricted to serpentine outcrops. And this variety, I've always thought, just from observations, might be very, very different in terms of not necessarily a subspecies of Quercus dorata variety gabrielensis, and which is found only or primarily in the San Gabriel Mountains. And it's got a restricted distribution, and I haven't figured out why it's restricted, whether it just hasn't gotten further, but that's something, many open questions. And then garryana variety breweri, I didn't get enough samples for this study, and I'm disappointed, but at least it's in the larger phylogeny that Andy and Paul and others have been working on. And then John Duckeri is Ducker's oak, and Quercus pacifica island oak. Now, I have to say, over the years, everyone says, how would you ever want to study the shrub oaks? They're such a mess. They're impossible to tell apart. No one's really sure. And I'll have some people say, oh my gosh, they're really not separate species. And others who swear they can tell one from another. And I'm not particularly good at telling them apart, except that if I go into an area, if I go in a particular region, I do pretty well. But as soon as I start moving around, they're so variable, I really have to work hard to do it. So as a geneticist, I wonder, well, if you can't tell what they are on the outside, surely on the inside we should be able to get it. So that it's always been a curiosity of mine to understand them better. And for one of the projects I've been doing for years in Southern California, understanding the shrub oaks is essential to understanding any of the population genetics of them separately. Here's, now these, Joaquin, and what he did is he used species distribution methods. And by the way, I am, for the sake of telling a story, going to skip lots of methods. And if you want to hear them, I'd be happy to explain every one of them. I can tell you any one of these parts could be a whole talk, but I want to tell a bigger story. So I'm skipping the methods today. But so we use species distribution models, and we paired them up so that you could see Quercus barbadofolia and Quercus dorata. I think that's a pretty generous species distribution model, and I suspect the reason it's so generous is that it doesn't take into account serpentine soils. So really what it would be is patches within it. And the other is my colleague pooled the variety gabrielensis with dorata, and I'm not sure. That's to me a hypothesis. And so in my later work with the sequence data, we separate them so we can actually test that hypothesis. But he just decided it was easier to pool them, so his maps do that. But the point is, barbadofolia really does have a wide niche, very wide distribution. This has a wide distribution, but within it, it's very specialized only on serpentine soils, and down here, I'm not sure. So widespread versus restricted. Then we have these two pairs. Cornelius Mulleri and John Tuckeri, where they both have, sort of narrower distributions, and I don't recall what they were calling this before they called it John Tuckeri, but it was only recently described as a separate species. And they kind of have disjunct distributions, if you see, and you almost wonder, whether or not these could be related, but they've both got smaller distributions. And then this pair, Damosa, is the coastal scrub oak. That people often before were confusing with barbadofolia, and then here's the island oaks, which are only on the Channel Islands, and you'll see why we put them together, but they're certainly close geographically. All right, now, this is where I'm going to start with telling you about using microsatellite data. So the advantage of microsatellite data is it's less expensive, and you can study lots of individuals. So we had about 20 microsatellites for this work. And when we pull them, we'd only pull populations that have at least 5 individuals, 16 nuclear markers. And what we did is we just looked at the rooted tree. So now we're just using the microsatellite data. And it looks like a pretty easy story here, which is that it's not surprising. Here's barbadofolia spread throughout. You can see dorata, that leather oak that's on serpentine soils. It comes next to it. People have often thought they're related. And then down here, Cornelius Mulleri. It comes off of barbadofolia. This is an unrooted tree. John Tuckeri. And then the 3, look at Damosa, which is that coast shrub, and the island shrub are right next to each other. So a pretty clean tree when you just look at neighbor joining with microsatellites. And a pretty good case that all of these. Now let's look at chloroplast. The reason to look at chloroplast is because we know that with chloroplast you can really retain information about prior integration at any point in time, because the chloroplast is maternally inherited. So if your mother was ever of another species, you're going to still retain that information. And when we look at that, we had 5 chloroplast markers, and same method, but you can see the tree is much more complicated. So you've got a lot more history of at some point in the time. Now with microsatellites, it's not as easy to figure out what point in time, but a lot better sense that hybridization has actually been very important in the history of these species. Now, there was more events, but the nuclear genes kind of got selected out, or are they just smaller number, but we don't get to pick them up? So, so from using this now, so this method, notice we did about 1,200 individuals. That's good sample sizes, good for population genetics, and it allows us to repeatedly sample, but with a limited number of markers, but we can sort of make these conclusions. The nuclear markers suggest why or indicate why we are able to maintain those separate species identities. And when you remember that figure, there's the Demosa and Pacifica. They came out next to each other, but they're very separate, so that would suggest geographical barriers are important. And when you have Berberifolia versus Dorata, which I separate, you can argue for ecological isolation. They're right side by side, but one's in serpentine soil and one has general soils. Then, of course, the chloroplast markers. So now let me talk about sequence data. Here we have the opposite kind of situation. For this study, we looked at 11 taxa and only 65 individuals across all trees and shrubs. Very expensive method. For a lot more money, you get to sample a lot fewer individuals. On the other hand, you get a lot more markers. So we went from 17 markers to just under 90,000 SNPs. And for those of you that do, we tried to use the same method that the other phylogeny is using, and that we use RADseq, and rather than run it in my lab, we actually send it to the same laboratory, so which turns out to be really advantageous, I think, for sharing our data. The biggest difference between us is I have a reference genome for Valley Oak, so I'm calling, the I is I should be, Sorrel is calling all of our SNPs with respect to reference genome, and we get pretty good rates of data across all individuals. So we're, and here is just the range. So we instead sample throughout the area, but very few individuals. Here's the tree, and I'd rather you not, this tree, I am so cautious about it that anyone that takes a picture and cites it, I will, but I want to just point out, so Valley Oak comes out as ancestral, and it's a pretty good clade, except we've got this Garryana in there that I don't know, and I don't know if there's integration there. We need to analyze a lot more, or the Garryana really is embedded in Valley Oak. I suspect it's integration. Really, look at this mess. That's all those different. So what I'm going to do is, before I go and publish this, I'm going to double validate, check the species identification, because what people tend to do is, they just sort of call it the species that's supposed to be there, and we've really got to verify these. So this is really, really preliminary data, but tell you, based on calling it just what you think you see, you've got a big mess, and this is a problem. Yet you get something like leather oak, that serpentine soil, and it clusters together pretty nicely. And then you've got scrub oak, island oak is by itself, blue oak or Douglasii has got its own clade. Look at that, looks good. And Engelmann oak, that Mexican oak, looks good, except it's related to this individual here. So we've still got to look at the role of integration in this tree. I'm going to start zooming. So we do see this evidence of isolation. One is in island oak, which has got a geographic barrier, and one in leather oak, which has an ecological barrier. Other shrub species are not found in distinct clades. They're still a mess, but I want to, we've got to double check all the species IDs on that. And the tree oaks tend to be show less integration. And especially Valley Oak, which every time they talk to me, they go, oh, how can you study it? How much hybridization? And you can see they're coming out separate, and I can find hybrids, but it's easier to find individuals with less. Now I'm going to have to go to this. I've got 5 minutes, so I'm going to just show you pictures to show you I do know what leaves look like. And I'm going to then skip through and just tell you why I did this study. I wanted to look at the processes of integration and hybridization. And we have done this both with morphological markers, microsatellite data, but I want to show you something very interesting. And skip through this because they've told me I've got 5 minutes, so I'm going to get to the genomic data. Here we used what's called the RNA data. That's the genes that are expressed, okay? So we're just looking at genes that are expressed in the study, and then we called the SNPs the variation. Using that, for this data, we had about 912,000 SNPs in the study, and 24 individuals. Again, you get lots of precision. And when you do add mixture, it says, yes, you've got 3 species for these 24 individuals. We deliberately, when we sampled these 24 individuals, we went to sites that we thought that we'd find pure individuals, and we went to sites that we expected to find hybrids. That was the intention. And so this should have a mix of each. And you can see we have lots of pure. We also have a few what could be, just first generation hybrids, and then a little peaking of Engelmann genes showing up in the Cornelius molari. And now that's for wide. Then we decided, since I've always thought one of the wonderful benefits of integration is that you can spread favorable alleles. So we looked at drought-resistant alleles and asked, are they showing the same pattern of movement across genes as if you just pulled all of them? And this is the result for that. Same order, same rank, and if you just base this, now we've got 1,900 SNPs. So if you say, oh, that's a lot less data, well, 1,900 SNPs is a lot more than 18 microsatellites. So it's a fairly powerful method. And you can see lots of integration, and you can see the patterns are very different. So let me just put them by each other. Those are the same rank order, and you see some pure individuals became, incorporated genes. Something based on all the SNPs looks completely like a Muller's oak, now actually has a lot of Berbertifolia drought-resistant genes. So the genes, the drought-resistant genes, are expressed in these individuals very differently than just random. So yes, my hypothesis that this is a good way for genes to move that might be beneficial for local adaptation, the mechanism is supported here. So, summary of insights about integration. Species have separate genetic identities. Morphological variation is greater than genetic. That's a part I skipped over. And integration is enhanced in genes associated with response to climate. So I'll give you what I'm going to call preliminary conditions. I mean, I'm just beginning to get in to this data, and my data aren't even completely analyzed, but I'm sharing with you them anyway, because I have colleagues here. I'd like their feedback. But my conclusions are, yes, oaks share genetic material across species due to historical and ongoing integration. I would argue, without showing you the data here, that ongoing integration is more common in some species sets than others. Speciation is shaped in oaks, is shaped both by reproductive isolation, the traditional mechanism, but also by ecological isolation. And species classification of several shrub species still in question. Do I have those multiple species in the same clusters because of integration and they're just moving around? Or are they really not separate species? And I don't yet know about that. And finally, Quercus lobata and Fagaceae, both of them show little integration. But Quercus engelmanni shows quite a bit, which is why I think it's a wonderful study system for trying to look at the process of integration. And I think Quercus engelmanni shows a lot of integration for 2 reasons. One is because it's recently arrived in California, it really has no isolating mechanisms. And it may be also the frequency of fires where you're constantly getting disturbance may create the opportunity. With that, there's a bunch of acknowledgments, and this is the tree that I'm going to immortalize when we sequence the Valley genome. Thank you very much.